Iris flower dataset analysis and predictions. Discovering patterns in famous flower data. Classification, visualization, and real-world insights. See how analysis leads to predictions. No coding experience needed. Running the cell now. What is the Iris dataset? A classic dataset for data science. Measures for features of 3 flower species. Used to learn classification and data mining. Easy to visualize and great for beginners. Running the cell now. Let us keep our notebook clean by hiding warning messages. Warnings do not mean errors, but can distract beginners. Suppress warnings for a clean output. Import warnings, warnings.filter warnings, ignore. Import numpy as np. We set the random seed to 42 for reproducibility. Running the cell now. This code sets a fixed random seed to ensure that any random operations in the Iris dataset exploration are reproducible, which is crucial for consistent results during experimentation or teaching. Although it produces no visible output, this step underpins the reliability of subsequent analyses and visualizations in this lesson on the Iris dataset. Let us download and load the Iris flower dataset from a public URL. We use Pandas, a popular data library, to read the file. We will check the shape to see rows and columns, and preview a few records. Data setup. Import Pandas as PD. URL equals https://raw.github-content.com/uiuc-cse/data-fa1 df equals pd read_csv URL. Print df.shape. Print df.head 3. Running the cell now. The output shows the Iris dataset has 150 rows and 5 columns, confirming it includes measurements for 150 flowers and their species labels. The first 3 rows illustrate the key features, sepal and petal dimensions, along with the species, which is essential for understanding the structure of this beginner-friendly classification dataset. This setup helps us get familiar with the data before exploring patterns and classification tasks. You should see the number of rows and columns, followed by a few lines of flower data. Each row is a different flower measurement. Before analyzing, let us see what features or columns our data has. What columns are in our dataset? Print df.columns.tolist. Running the cell now. This output shows the 5 columns in the Iris dataset, for measurements of the flower's physical features and 1 species label. Noticing these columns helps us understand what data we have for analysis and sets the stage for exploring how these features can classify the 3 species. This step is key for getting familiar with the dataset structure before diving into visualization and modeling. These features describe flower size and type. The species column tells us which flower it is. We want to make sure all data is present and check info. Missing data can cause errors during analysis. Basic info and missing value check. Info equals df.info. Missing equals df.isnal.sum. Print missing. Running the cell now. This output shows the Iris dataset has 150 entries and 5 columns, with no missing values, confirming the data is complete and ready for analysis. Notably, the 4 numeric feature columns and 1 categorical species column align with the dataset structure, setting a solid foundation for learning classification techniques. Checking data integrity here ensures smooth visualization and modeling in later lessons. No missing values means our dataset is ready for analysis. If you ever see non-zero numbers here, you have to fill or drop those entries. Describe gives basic summary statistics for each number column. It shows averages, minimums, and maximums to help us understand ranges. Show summary statistics. Print df.describe. Running the cell now. This summary table shows key statistics for each feature in the iris dataset, including the count, mean, and range of measurements. Notice how the sepal and petal lengths and widths vary, giving us an idea of the typical flower size and diversity across the three species. Understanding these stats helps us grasp the dataset scale before moving on to classification and visualization. Note the differences in means and ranges between features. This helps us guess which measurements separate the flowers. Visualizing the data. Pictures reveal hidden patterns fast. We can quickly spot relationships and trends. Visualization helps us choose analysis paths. Running the cell now. Let us make a simple scatter plot to compare two important features, petal length and petal width. We use dot colors to represent different species. This can show separation. Plotting petal length versus petal width. Import matplotlib.pyplot as plt. plt.figure, fig size sad smiley 6:4. For species in df, species.unique. plt.scatter, df, species, equals species, petal underscore length. df, species, equals species, petal underscore width, label equals species. plt.xlabel, petal length. plt.ilabel, petal width. plt.title, petal size by flower species. plt.legend. plt.show. Running the cell now. This scatter plot visualizes petal length against petal width, with points color-coded by species, revealing distinct clusters for each flower type. Notice how the separation in petal size helps us quickly identify species differences, guiding us toward meaningful features for further analysis. This demonstrates how visualizing data uncovers patterns that inform our next steps in exploration. You should see groups of dots in different areas of the graph. Some species only appear in certain sections. That is a strong hint we can predict the species with these features. Histograms show how many flowers fall into each value range. Let us look at the spread of sepal lengths. Histogram of sepal length for all flowers. df, sepal underscore length, dot hist, bins equals 20, color equals sky blue, edge color equals black. plt.xlabel, sepal length. plt.ilabel, frequency. plt.title, distribution of sepal lengths in dataset. plt.show. Running the cell now. This histogram shows the distribution of sepal lengths across all flowers in the dataset, revealing a clear concentration around certain values. Notice the shape. It helps us quickly see the most common sepal length ranges and whether the data is symmetric or skewed. Visualizing this guides us in understanding the data's characteristics before deeper analysis, such as comparing species or identifying outliers. Notice the spread and where most flowers cluster. If any species has unique lengths, this could separate groups. A pair plot lets us view every feature combination at once. Using color, we can compare how groups are spread out. This is a favorite tool for initial exploration. Pair plot for relationships among all features. Import seaborn as SNS. SNS.pairplot, df, hue equals species, diag underscore kind equals hist. plt.show. Running the cell now. This pair plot displays scatter plots and histograms for all feature pairs, colored by species, helping us visually compare their distributions and relationships. Notice how different species form distinct clusters or overlap in certain feature combinations, revealing key trends and separations. This visualization guides us in selecting which features to focus on for more detailed analysis and modeling. If you see clean separation with color, those columns could help predict species. Spotting overlapping areas can show where confusion happens. Preparing the data for prediction. Machine learning needs numeric inputs only. We will split our data into train and test sets. We use the train data to learn patterns. Test data lets us check how well our future predictions might do. Running the cell now. Many machine learning tools cannot use text categories directly. We convert species names into numbers using label encoding. Convert species names to numbers. From scikit-learn.preprocessing import label encoder. L equals label encoder. df species underscore num equals l.fit underscore transform df species. Print df species underscore num dot head 6. Running the cell now. This output shows how the species names, which are categorical text data, have been converted into numeric labels using a label encoder. Notice that all set entries are mapped to 0, preparing the data for machine learning models that require numbers instead of text. This step is essential for enabling algorithms to process and learn from the species information effectively. Now the species underscore num gives each species a unique number. We can use these numbers for modeling. Splitting the data prevents overfitting. We use 80% for training and 20% to test results. Random underscore state keeps splits the same each time for learning. Split data into train and test sets, 80 to 20 split. From scikit-learn.model underscore selection import train underscore test underscore split. Features equals df.drop, species underscore num, axis equals 1, columns. X equals df, features. Y equals df, species underscore num. X underscore train, X underscore test, Y underscore train, Y underscore test equals train underscore test underscore split, X, Y, test underscore size equals 0.2, random underscore state equals 42. Print, train, X underscore train dot shape, test, X underscore test dot shape. Running the cell now. This output confirms that the data was successfully split into training and test sets, with 120 rows for training and 30 rows for testing, each having for feature columns. This split ensures we have enough data to train our model while reserving a separate set to evaluate its future performance. Noticing the shape helps verify that 80% of the data is used for learning patterns, aligning with our intended 80 to 20 split. Your output shows how many rows are for training and for testing. This helps prevent our model from memorizing instead of learning. Predicting species with k nearest neighbors. A simple model that checks which flowers are closest. If neighbors agree, predicts their type. KNN is popular for beginners and works well here. Running the cell now. We train a simple KNN model with k equal to 5. Accuracy shows the percent of test flowers predicted correctly. Fit a KNN classifier. From scikit-learn.neighbors import kneighbors classifier. KNN equals kneighbors classifier, n underscore neighbors equals 5. KNN.fit, x underscore train, y underscore train. Score equals KNN.score, x underscore test, y underscore test. Print, KNN accuracy, score of 0.2%. Running the cell now. The output shows a perfect 100% accuracy, meaning the KNN model correctly predicted all flower species in the test set. This highlights how well KNN works here by leveraging the closest neighbors to classify each flower accurately. It demonstrates the power of this simple but effective method for species prediction. You should see a high accuracy, often above 90%. That means the features can predict most flowers correctly. A good dataset will help any model. Let us see if just petal size is enough for prediction. This tests if fewer features can give similar results. What features matter most? Try with fewer features. X underscore train underscore small equals X underscore train, petal underscore length, petal underscore width. X underscore test underscore small equals X underscore test e petal underscore length, petal underscore width. KNN2 equals kneighbors classifier, n underscore neighbors equals 5. KNN2.fit, X underscore train underscore small, Y underscore train. Score 2 equals KNN2.score, X underscore test underscore small, Y underscore test. Print fknn with petals only, score 2.2%. Running the cell now. This output shows that using only petal length and petal width as features allows the k-nearest neighbors model to perfectly classify the flower species, achieving 100% accuracy on the test set. Notice how reducing the features to the most important ones did not harm performance, highlighting that these petal measurements are key predictors. This reinforces the lesson's point that KNN can effectively predict species by focusing on the closest neighbors based on the most relevant features. Notice how the score changes. If accuracy stays high, just these features separate species very well. Try predicting a flower type using real measurements. Type real or sample numbers when prompted. For example, use 5.1, 3.5, 1.4, 0.2. This simulates making a prediction in practice. Predict the class of a new flower, interactive. S_length equals float, input, enter sepal length in cm. S_width equals float, input, enter sepal width in cm. P_length equals float, input, enter petal length in cm. P_width equals float, input, enter petal width in cm. Sample equals S_length, S_width, P_length, P_width. Pred equals knn.predict, sample, 0. Print f predicted species, l.inverse_transform, pred, 0. Running the cell now. This output shows the result of our k-nearest neighbors model predicting the species of a new flower based on the user's input measurements. Notice how the model takes the 4 features, sepal and petal dimensions, as input to find nearby flowers in the dataset and then predicts the most common species among those neighbors. Here, it predicted setosa, demonstrating how KNN uses closeness in feature space to classify new samples. You can repeat this with different numbers to see how the prediction changes. Try values from different species you saw in earlier samples. Try it yourself, mini exercise. Use the interactive prediction cell again. Try flower measurements from each species group. Can you trick the model? What happens if you enter values out of the shown ranges? Running the cell now. Pause the video and try this activity. Feel free to adjust different feature values for practice. A confusion matrix shows exactly which species were confused. Misclassified rows point out what the model struggles with. Confusion matrix for detailed performance. From scikit-learn.metrics import confusion_matrix, confusion matrix display. Import numpy as np. y_pred equals knn.predict x_test. cm equals confusion_matrix, y_test, y_pred. disp equals confusion matrix display, confusion_matrix equals cm, display_labels equals la.classes_. Dispplot, kmap equals blues. plt.title, knn classification confusion matrix. plt.show. Running the cell now. This confusion matrix shows how well the KNN model classified test flowers by species, with darker blue squares along the diagonal indicating correct predictions. Notice any off-diagonal values reveal where the model confused one species for another, highlighting its limitations when input values are close or out of typical ranges. This helps connect the lesson's goal of experimenting with flower measurements to understand how model predictions can be tricked or misled. Diagonal cells are correct predictions, off-diagonal cells are mistakes. If most numbers are on the diagonal, the model is doing very well. Let us try a different classifier, the decision tree. Decision trees split data by rules and can work well on small datasets. Can other algorithms do better? Try a decision tree. From scikit-learn.tree import decision tree classifier. Tree equals decision tree classifier, random_state equals 42. Tree.fit, x_train, y_train. Tree_score equals tree.score, x_test, y_test. Print f decision tree accuracy, tree underscore score of 0.2%. Running the cell now. The output shows the decision tree model achieving 100% accuracy on the test set, indicating it perfectly classified the flower species based on their measurements. Notice this contrasts with previous models and highlights how different algorithms can handle the same data with varying success. This experiment encourages you to test predictions with flower measurements, even unusual ones, to explore the model's behavior and robustness. Compare this score to KNN. Which model do you find more reliable on this dataset? Decision trees can be shown as a picture. Each box shows a split rule and how the data divides. Here we limit the levels to keep the plot readable. Visualize the actual tree structure, simplified. From scikit-learn import tree as scikit-tree. plt.figure, fig size sad smiley 10:6. scikit-tree.plot underscore tree, tree, feature underscore names equals features, class underscore names equals la.classes underscore, filled equals true, max underscore depth equals 2. plt.title, decision tree structure, first 2 levels. plt.show. Running the cell now. This decision tree visualization shows the first 2 levels of splits based on key flower measurements, highlighting how the model separates species using feature thresholds. Notice how the tree uses specific measurements to funnel samples toward different species predictions, which helps in understanding why certain inputs might trick the model. This connects to the exercise by illustrating the decision boundaries that can be explored and challenged with new, possibly out-of-range values. Notice that splits are based on feature values, like petal or sepal sizes. If you understand the rules, you can almost hand classify new flowers. Next steps and practice. Try other classifiers on this dataset: random forest, logistic regression. Practice more plotting with Seaborn or Matplotlib. Can you create your own small flower dataset and predict manually? If you like this video, subscribe and comment below for more beginner-friendly data lessons. Running the cell now.